glycan, how it's cross-linked, and what it's made out of. So I really want you to understand a lot about peptidoglycan, mainly because it's a source of antibiotic attack. It's the place where antibiotics can attack the bacteria. All right, so we talked about it's made out of mainly a single line of N-acetylmuramic acid and N-acetylglucosamine. All right, it's just a polymer. Remember, polymer means many, so it's a lot of these monomers linked up to form a long polymer. All right, and then these long chains are cross-linked by these little short pieces of protein, polypeptides. So each one of these balls is an amino acid. All right, 4 amino acids, 1, 2, 3, 4, and then they are linked by 5 amino acids. Now look here, what kind of cells are those in? Positive or negative?
Positive.
Positive, there you go. So you may guess this is going to have something to do with gram-positive bacteria using a certain kind of antibiotic to kill them. All right, so we went over this last time, but here it is again. Then remember we talked about the teichoic acids. They're kind of like the rebar in the concrete, right? So teichoic acid is here, and then here you have the long chains of NAM and NAG. Like I said, my little mnemonic devices don't nag me because I was in NAM. So NAG, NAM, NAG, NAM.
Those circles are still amino acids, though?
Yes. No. These circles are the NAG and NAM. NAG, NAM, NAG, NAM, NAG, NAM.
Oh,
Right
yes,
here
okay.
are the amino acids, right? 4, 4, 5. Linking those together. So if you break that link, you're going to pop the cell open, which is how antibiotics work, some of the antibiotics. Okay, so difference between gram-positive and gram-negative. The main difference that lets that crystal violet stain stay in there is this right here, the peptidoglycan and teichoic acid. It's thick right here, so that's just going to keep that crystal violet stain from coming out. Pretty straightforward concept. Okay, so then in contrast, here's the gram-negative wall. It still has some peptidoglycan in it, but notice how thin it is, right? You just have one thin little layer of peptidoglycan. All right, and then another difference is you have this lipopolysaccharide here. Okay, you have a porin channel that goes out from the inner layer to the outer. All right, and then you still do have this membrane that's the phosphobilipid layer, cytoplasmic membrane. All right, and you have a space in here. This is called the periplasm. All right, remember we talked about E. coli. I'm not going to try to say the number. What is it? H7?
What is H5N1?
No, that's a flu.
I remember something.
Okay, so it's,
0157.
there you go.
Are you kidding me?
Yeah.
Something
to that.
right now.
There you go. There's some letters in there, but
Yeah,
yeah. Okay, so the type of E. coli that causes intestinal disease is going to have this O antigen there. It's going to have endotoxin. All right, now how is it able to let the crystal violet out? It has this really thin layer of peptidoglycan, so when you decolorize it with the acid alcohol, it's going to let the crystal violet just come back out. All right, so gram-positive are going to be what color?
Purple.
Outstanding, and gram-negative will be
Pink.
pink because they're counterstained with the safranin. Okay. Bilayer made from lipopolysaccharide. And again, the reason that the gram-negative are really bad is your body is going to try to attack them and ramp up a huge immune response against that lipopolysaccharide. So again, if you have a small level, it's going to be okay because it's going to give you a good immune response. But if you have a lot, unfortunately, you can have a deadly immune response and end up dying from that immune response. Okay, so the lipopolysaccharide is also called endotoxin. All right, so endotoxin is on the cell membrane. You also have exotoxins, which are excreted. So in this case, we're talking about an endotoxin. Okay, so the parts, again, are lipid A and the O antigen are the parts of that endotoxin. Okay, and again, there is a space between the cytoplasmic membrane, which is on the inside, and the periplasmic membrane. So in between there, you have the periplasm, right? You've seen that little word before, plasm, right? The cytoplasm. So here, this jelly stuff is in between the cytoplasmic membrane and the outer membrane, and it's called the periplasm. All right, and what goes on in the periplasm? Exported proteins accumulate unless they are moved across the outer membrane.
I'm sorry, what happens in there? In the periplasm?
These proteins are going to be accumulated unless there is something to move them out. Okay, so we just said this, but again, this peptidoglycan is going to be a good target because it's only going to be found in the bacteria cell walls. So we do not have peptidoglycan, so if you destroy peptidoglycan, we don't care. You're not going to have any negative side effects from that. Okay, so what happens, basically, the cells are just going to weaken and then they're going to blow up. Lyse. Cell lysis. The peptidoglycan layer is going to be so weak that the cell just opens up. Alright, so again, it's going to prevent cross-linking of the glycan chains. The NAM, NAG, NAM, NAG, those proteins in the middle are not going to link to each other. Okay? And again, of course, they're usually more effective against gram-positive than gram-negative. Who can tell us why penicillin is more effective against gram-positive than gram-negative? More specifically, please. There you go. Outstanding. Okay, the peptidoglycan is going to be thick in the gram-positive, so it's going to have a larger effect. Okay, another question. Which is going to have a bigger effect? Penicillin on growing cells or penicillin on static cells? Growing, there you go. Because that cross-linking is still going on and the cell wall is still being formed as the cells are growing. All right, so we have just mentioned penicillin. We also have in our wonderful bodies an enzyme called lysozyme. Lysozyme, where do you find lysozyme in your body? I'll give you a hint.
Tears.
In your tears, very good. Okay, also in your saliva. Lysozyme is going to, again, break down the bonds linking that glycan chain. Okay, tears, saliva, and other body fluids. All right, so that's going to, again, break down your peptidoglycan molecule. Okay, we actually did this, crystal violet stain. It actually worked, yay. And it prevents the crystal violet from being washed out, the thick peptidoglycan layer, and that makes the gram-positive purple. Okay, then you decolorize, and again, the thin peptidoglycan of gram-negative is not going to keep that dye in, so the purple dye washes out, and the pink safranin will be retained in the gram-negative. Okay, we talked already about the different shapes of bacteria: coccus, bacillus or rod, spirillum, spirochete. Comma-shaped is Vibrio. Right, and then we have these weird guys here that don't have much of a shape at all. Well, they do have a shape, but they have no defined shape. They're kind of blobby. Right? How are they able to be so blobby shaped? What are they missing that other bacteria have? A cell wall. There you go. So without a cell wall, their cytoplasm is going to be able to flow more freely. Okay? So without a cell wall, will these things that attack the cell wall have any effect? No. So penicillin and lysozyme are not going to be effective against these bacteria lacking a cell wall. Okay, how are they able to stay tough and rough? Without a cell wall, they have sterols. So sterols in their cell membrane are what's going to give them that strength. Okay, archaea, we talked about archaea being the extremophiles, extreme hot, extreme cold, extreme salt. Okay, so why is that possible? They have a different molecule that's called pseudopeptidoglycan. Okay, and it's built from these sheets of flat protein or glycoprotein substances. All right. So outside the bacteria, you have slimy, icky stuff on the outside. We already talked about that in the biofilms, right? So it has a capsule, which is going to be this distinct gelatinous slime layer. It's composed mainly of glycocalyx, which is a sugar, right? A sugar shell, and some have polypeptides or short units of protein. All right, and again, like we mentioned, once they attach, they can grow as a biofilm, and unfortunately, this can result in tooth decay. All right, flagella. We have actually different types of flagella. We have some that spin, some that are whip-like. Right, then these are good microbiology words to know. Peritrichus, they're over the whole surface. Polar flagellum, just like we have one North Pole, it's a single flagellum. All right, and some have a tuft at one end or the other. All right, this is very interesting. The prokaryotic flagella looks like this. And we already reviewed the other day how hydrogen is used to create that gradient. And as the hydrogen flows down over the cell membrane, it's going to give energy. We mentioned it's used to make ATP. Well, it's also used to spin this flagella, right? So it harvests the energy of the proton motive force. Proton motive force is when those hydrogens are flowing across the cell membrane. Okay, remember we had a whole bunch of hydrogen on one side. All right, and they just flow past this thing. This is not obviously exactly what it looks like, but it's very similar. It's so cool. You have to look it up. It's a little molecular machine. The hydrogen flows across, and then the tail spins. Pretty neat. All right, chemotaxis. I'm going to show you chemotaxis. Okay, ready? I'm going to do chemotaxis now. All right. I smell some cookies. All right, no, actually, chemotaxis would be just like that. I smell cookies downstairs, and then I would just walk off, go downstairs, and get the cookies, right? I would follow that trail. All right, so what happens when there is no chemical gradient? Then you have that run and tumble thing. So you're going to go and then just kind of spin around. You ever play that game when you're a little kid? Spin around. Okay, let them go. Go. Spin around. Go back. Okay, that is the run and tumble mechanism when there's no actual chemical gradient to follow. All right, so bacteria are so amazing, they can also follow other kinds of gradients. All right, they can do, wrong way, aerotaxis. Aero means what? Air, right? So they can follow like an oxygen gradient. They actually can have metal and magnets on the inside. They can follow the Earth's magnetic waves. It can also follow heat, thermotaxis, and what do you suppose phototaxis is? There you go, they can move toward light. All right, so bacteria are actually pretty amazing.
So the taxes pretty much mean movement.
Think of a taxi,
Okay.
right? When you want to move in New York, you're like, taxi! Okay, so taxi is movement.
So as long as we can remember, obviously, aeromagnet, thermo, and photo, we can figure out
There you
because it's all
go.
the same, just a different
You got it.
Magnet feet or like, okay.
Okay, so little parts. We already talked about pili, but just to review, pili are like short little flagella. All right, they can attach. So the ones that are attaching, like attaching to a surface, are also termed fimbrii. Okay, then they can also move, right? Twitching motility. And they can glide. So if they're swimming or twitching, they're using their pili. Now, this is a really good one. There's also an F pilus. All right, so F pilus. You can have an F plasmid or not have an F plasmid. All right, so bacteria, they're sort of like male and female, but not really. All right, so some have the F pilus and some don't. But amazingly enough, we mentioned what a plasmid was. Can anybody tell me again what a plasmid is?
It's like jelly.
Oh, no, sorry.
That's plasma.
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 Isn't it still jelly?
Plasmid. Here's the word, plasmid.
Container.
DNA, there you go, outstanding. It is a circle of DNA. Okay, are you guys on the wanted list or anything? I say your name on YouTube, is it okay? Okay, so.
Okay, I'm gonna put on it, as long as I can't see it.
Okay, so let's see. We'll just call you Betty. Good job, Betty. Okay, so plasmids can actually go through the little F pilus. The F pilus. This is really bizarre, but one of my students gave me an onion ring that was shaped like an F pilus. I thought it would be too bizarre to keep it, but I was tempted. I was like, I should put this away. But so basically, think of an onion ring with a little extra thing off of it, right? Like this. All right, that is your F pilus. Okay, so the F pilus is going to go ahead and make a little tube like that, and then this plasmid is going to open up and just get squirted through here. Okay, so that's step 2. It opens and it goes through to the next bacteria. And then finally, it's going to be over in the next bacteria. Okay, and this guy might have more than one of these, so it can still have its F pili. All right, and now that this guy has the F plasmid, guess what? It now has the F pilus. Okay, so that's pretty cool. They can develop this pilus by transferring a plasmid, which of course has the DNA. The DNA has the genes that code for the protein to make this pilus. All right, unfortunately, what else can plasmids code for? You guys got it?
HIV?
No, that's a virus. We're talking bacteria here.
Oh.
What are bacteria developing lately that we do not want them to have? Resistance, there you go. So not only can you have F plasmids, you can also have R plasmids. R stands for resistance. Okay, so same idea here. This plasmid is going to be transferred, and unfortunately it can also be transferred to an entirely different species. So whereas you had one bacteria that had no resistance, now you can have another one. Another reason to be nice and clean, right? You don't want those bacteria combining together. All right, and here's a picture of that again. Okay, so this is what DNA looks like normally when it's just coiled up in the nucleolus. Not coiled up, but free in the nucleolus. All right, so chromosomes, they form a gel-like region, which is called, again, the nucleoid. So do bacteria have nuclei?
No.
Good job. They do not have nuclei. All right, and again, plasmids, circular, supercoiled double-stranded DNA. DS stands for double-stranded. And again, antibiotic resistance can be shared by the plasmid moving between two different bacteria. Now, we talked a bit about ribosomes. Here they are again. Ribosomes are where proteins are made, of course. We've already had that in A and P. They're going to link together the amino acids. Okay? And again, this S means how fast they're going to move. It's kind of a measure of their aerodynamic ability. All right? So if you have a test tube here, you put the ribosome in there, in some gel stuff, and then you put that test tube in a centrifuge. It's going to spin around, and it's going to go to the bottom. How fast it goes to the bottom is telling you what number it's going to get. All right, so here again, our new math, 30 plus 50 is 70. And how about in the eukaryotes? It's 40 plus 60 is 80. Okay, and it makes sense if you think about it, because before they were separate like this, and then when they join together, it's still going to be, you know, fairly aerodynamic if it goes this way. All right, and again, we do have antibiotics that are going to exploit this difference. It's much better to have prokaryotic being attacked than eukaryotes. Unfortunately, and this is something we're going to get to in a minute, is that our mitochondria actually have 70S ribosomes. So be thinking about that, or if you actually read your chapter, you know, what does that indicate? How in the world do our mitochondria have prokaryotic ribosomes? Think about that, see if you get to the answer. All right, then we have the cytoskeleton. Cytoskeleton is just a bunch of proteins that link microtubules and so forth, and they're going to, give the cell its shape. And again, cell division. Okay, storage granules. What's in a storage granule? Well, it's storage. It's basically just going to be a large, tightly packed together with polymers, such as starch. Okay, so glycogen, poly, beta, hydroxybutyrate. Okay, we actually get that from microbes. Okay, and I think we already talked about this, but again, gas vesicles are going to let the bacteria float up and down at a good level. So let's say that there was not enough oxygen, they could float up to the top of the lake, something like that. Okay, endospores. We talked about endospores, and what do they form? They form when the nutrient levels are not right. So like if there's not enough carbon, not enough nitrogen, they can form spores. And here's a picture of an endospore forming. Of course, eventually all this vegetative stuff is going to go away and you're just left with a spore. Okay, and grossly enough, they're found almost everywhere. So you go and get a sample of dirt, you're probably going to pick up some endospores. And this is important right here, especially for those who are going to be nurses. They are resistant to heat, desiccation, chemicals, UV light, and boiling water. So how in the world do you get rid of them? That too.
I read it. I know I read it.
Okay,
Something to do with
there you go.
Something
So
like kind of like
if you autoclave for a long time, you can kill them. Sporulation, again, starvation. It takes about 8 hours to form a spore. All right. these are going to keep out the lysozyme. So unfortunately, when you have a spore in your body, your tears and your saliva are not going to be able to destroy that spore. Okay, it's got a cortex around the outside, protected from heat. And then it also has proteins that are going to bind onto the DNA itself and protect the DNA. Okay, then it also has this calcium dipicolinate. Right, and that is also going to be protecting the DNA and not let it be destroyed. Okay, and then the germination is going to be triggered by heat or chemical exposure. Basically telling it, okay, the nutrient levels are supposed to be right again, so go ahead and start being vegetative. All right, and then here we have a slide that shows you some of the differences between animal and plant cells. What I really want you to emphasize is the difference between prokaryote and eukaryote. So we're going to take a pause right here and draw ourselves a beautiful eukaryotic cell.